CO2, and it is my honor to have a chance to present my work here. And today, my talk is to compare the photosynthesis in mesophyll cells and gut cells in Arabidopsis. So it is well known that the mesophyll carry out photosynthesis and export sucrose to support plant growth. And for gut cell, they control carbon dioxide intake and transpiration. And gut cells also have chloroplasts. So does gut cell chloroplast carry out photosynthesis has been a debate for decades. So now let's look at the photosynthesis in mesophyll cells. So in the chloroplast, there are photosystem 2 and photosystem 1, and electrons are extracted from water molecules, and then it will pass through the photosystem 1, and then is used to reduce NADP, the electron acceptors, into NADPH. And at the same time, the linear electron flow also create a proton gradient that can drive the generation of ATP. So the linear electron flow can produce both ATP and NADPH, and someone have worked out that their ratio is 1.286. And of course, there are also some cyclic electron flow that only generate a proton gradient and that can supply extra ATP. So to fix one CO2 molecules at the Kelvin cycle, it will consume 3 ATP molecules and 2 NADPH molecules. So the demand of ATP to NADPH by carbon fixation is 1.5. But the supply of ATP to NADPH by the linear electron flow is 1.286. So it seems that there is a shortfall of ATP. So if ATP is insufficient, then it will cause the accumulation of NADPH, and that means that would be insufficient NADP, and this is the electron acceptor, and this will slow down the linear electron flow. So the ratio of ATP and NADPH has to be balanced. So the question is, how does plant chloroplast obtain additional ATP? So of course, cyclic electron flow can generate ATP, but how about ATP importation from cytosol? So in this 2015 Nature paper, they studied diatoms, and in the diatoms, this is the chloroplast. The surplus NADPH can be converted into malate, which is then exported to the cytosol, and which is then import in the mitochondria to release NADH. And this can be used by the mitochondria electron transport chain to produce ATP, and the ATP can be exported to the cytosol, and then the key point is it can be imported into the chloroplast to supply extra ATP for the Kelvin cycle. So in diatoms, cytosolic ATP can enter chloroplast to supply extra ATP for the Kelvin cycle. But how about in plants? So half a century ago, a paper was published. The title of this paper was Adenine Nucleotide Translocation is Spinach Chloroplast. And this paper has been cited more than 200 times. And its conclusion is that ATP can move in and move out mature chloroplast freely. And it was suggested that this, at the daytime, it can supply ATP to chloroplast to support photosynthesis. And in this 2011 review paper, it says that chloroplasts during the night rely on the import of energy in the form of ATP via the NTT. The NTT is the ATP transporter on the chloroplast membrane. And our group have adopted an ATP sensors, which is a fluorescence protein called AT1.03. This protein actually has 2 fluorochrome, which is linked by an epsilon subunit, which is able to bind ATP. So if there is a higher concentration of ATP, then it will bind here, and then these 2 fluorochrome, their distance would become closer, and then there will be a Foster resonance energy transfer between these 2 fluorochrome. So this fluorochrome can be excited by 435 and emit at 475. And if there is a flat transfer, And this photochrom ambinus can emit at 5 to 7. So if we use the same excitation wavelength and monitor these two emission wavelengths and then look at their ratio, then the ratio actually is a proportion to the concentration of ATP. And then so in this region is quite linear, and then there is an upper limit of 1.4 millimolar ATP. So this sensor was developed by Professor Imarura, and then we introduced this into Arabidopsis. The advantage of using this ratiometric sensors by looking at the ratio is that it is independent of protein expression level. That means that if we express these proteins in different cells and in different organelles in Arabidopsis, although the sensor concentration could be different in different cells or in different organelles, but we don't need to worry about that. We just look at the ratio. And we also introduced another pH sensors called CPYFP. And this one can be excited by 205 and 488 and emitted at 520. And then we can look at the ratio of the emission by these two activation wavelengths. And we can see that its ratio also follow the pH change. So we can use this as a pH sensors. So the advantage of using this ratiometric fluorescence protein sensor is that we do not need to carry out plant extraction. The measurement is in planta. We can look at individual cells, you can look at individual organelles, and we can also carry out real-time monitor. So you can see that we have also introduced an NADPH sensors into pollen plastids, and you can see that different pollen plastids have different NADPH concentration. So we have introduced the AT1.06 ATP sensors into chloroplast and cytosol of Arabidopsis, and we have also introduced the pH sensors into the mitochondrial matrix. And then we monitor the change of this sensor by elimination or by application of different inhibitors. So in the 10 days old seedlings, we look at the ATP concentration in the plant, and blue color means low ATP concentration. Red means high ATP concentration. From here, you can see that the plastids in cotyledon, hypocotyl, and root, they are blue color. But in the cotyledon cytosol, is it red, and this is orange, and this is a bit green. So that means that in 10 days old seedlings, the cotyledon cytosol has very high ATP concentration, which exceeds the upper limit of the detection limit of the sensors. And in the hypocotyl, is it in the detection range? And in the root cytosol, is it in the detection range? It was about 0.7 millimolar in the root cytosol. However, in the plastid, it is very low, okay, around 0.2 millimolar. So we have a question: why stromal ATP concentration is lower than cytosolic ATP concentration? And there are two possibilities. One is inefficient importation of ATP, and ATP consumption in chloroplast is tremendous. So we try to look at younger seedlings. We look at 3 days old, 4 days old, and 6 days old younger seedlings, and we can see that the ATP concentration in the plastids decrease when the seedlings mature. So in Arabidopsis, there are entity transporters on the chloroplast membrane that allow the antiport of ATP and ADP and PI. So, if the import of ATP will followed by the export of ADP and PI to the cytosol. And in Arabidopsis, there are two entity genes. Entity 1 is mainly expressed in the vein, whereas entity 2, you can see that it's highly expressed in the cotyledon, day 1, day 2, day 3, but the expression is downregulated in day 6. And the entity transporter is only expressed in the root and the young leaf. So our hypothesis is that in mature chloroplasts, they downregulate NTT transporter so as to restrict the importation of ATP from the cytosol. To examine this, we have isolated chloroplasts carrying the sensors from 4 days old, 5 days old, 10 days old, and 20 days old seedlings, and then we add exogenous ATP. If the ATP can get into the chloroplast, the Fred ratio will increase. And then we can see that ATP can get into 4 days old chloroplast, 5 days old chloroplast, but not 10 days old or 20 days old chloroplast. So this observation fits with the gut staining. So our data show that actually for mature chloroplast, the ATP outside of the chloroplast cannot get into mature chloroplast because of the downregulation of NTT. So why our data contradict with this paper? We try to look at and examine their experimental procedures. We can see that first, he isolates chloroplast, mature chloroplast from spinach, and then he added radioactive ATP to the isolated chloroplast. And he assumed that ATP has been input into the stroma, and then he washed the chloroplast to remove the radioactive ATP outside of the chloroplast and then add co-ATP. And then he counts the radioactivity in the supernatant, assuming that the co-ATP here could exchange the hot ATP inside the chloroplast. However, NTT actually is an exchanger of ATP to ATP. If you look at his own data, he showed that addition of ATP, 34%, addition of ADP, only 4.1%. But if the radioactive ATP is indeed get into the stroma, then addition of ADP should be more efficient than ATP. But this was not the case. So that means that his data actually showed that there was no ATP translocation, but the radioactive ATP he added only absorbed on the surface of the chloroplast, so that when he added co-ATP, the radioactive ATP just dissolved from the outside or the membrane of the chloroplast. So we believe that the mature chloroplast does not import cytosolic ATP. Then we try to, for 10 days old seedlings, we try to use light to eliminate the cotyledon. And we can see that the light, when we turn on the light, the stromal ATP increased from about 0.2 millimolar to 0.35. But when we turn off the light, within 30 seconds, it dropped to a low level. We turn on the light, it increased again. We turn off the light, it dropped again. So that means that in the dark, the chloroplast consume large amount of ATP. Then we try to isolate protoplasts that carry the ATP sensors in the cytosol, and then we transfect a vector containing NTT1 or NTT2. And after overnight incubation, we can see that the cytosolic ATP in the protoplast drop to a very low level if we transfect them with NTT1 or NTT2. And this does not happen if we only transfect the vector. So that means that if mature chloroplasts still express high concentration of NTT transporters, the chloroplast will consume a large amount of cytosolic ATP at night, and is not energy efficient because within 1 day, within 24 hours, there are about 8 to 12 hours of darkness, depending on where we are. And then we also try to constitutively express the NTT1 transporters. And then we find that if we grow the NTT overexpression line with the vector line in long day, they have similar plant diameter, but if we grow them in short day, okay, short day, overexpression of entity express transporter will result in a smaller plant. So that means that this also explains why mature chloroplasts should downregulate NDT transporter, because it will waste the cytosolic ATP at night. So our major findings include, in mesophyll, the chloroplasts consume large amount of ATP in the dark. And unlike unicellular diatom, cytosolic ATP does not enter mature chloroplasts of Arabidopsis thaliana. Mature Arabidopsis chloroplasts downregulate NDT transporter to reduce energy expenditure in the dark. So why is it different from diatoms? Is it because diatom is unicellular? They just take care of themselves and they do not need to export sucrose. But for Arabidopsis, is it multicellular? And the major function of mesophyll is to export sucrose. So it has to reduce its energy expenditure in the dark. So NDT transporters is only expressed in the proplastids during chloroplast biogenesis, because at that time, the proplastids cannot carry out photosynthesis, and ATP is needed for protein translation, protein import to build the chloroplast. And so for mature chloroplast, because there is no NDT transporter, so in the dark, mesophyll chloroplast need to carry out plastidic glycolysis in stroma to meet its energy or ATP demand in the dark. Then using our ATP line, we can also test different inhibitors. And you can see that using rotenone and TTFA, which is the complex 1 and complex 2 inhibitor, we can lower the cytosolic ATP to about 0.8 to 1 millimolar. And if we use oligomycin, then mitochondria cannot generate ATP, and then the cytosolic ATP disappear. So this proves that mitochondrion is the major source of cytosolic ATP in the dark. And here, if we use rotenone to lower the cytosolic ATP to a detectable range, and we can see that this is the light, 30 seconds, 1 minute, and then we can see that the light can increase the cytosolic ATP in a minute. And this is DCMU dependent. And if we inhibit the mitochondria to generate ATP, then if we apply light, we can still see increase in cytosolic ATP, but this could be due to glycolysis. So we have introduced the pH sensors into the mitochondrial matrix, and we can see that if we apply light, it has alkalization. We turn off the light, it will drop. We turn on the light, it increases, and turn off the light. So that means that elimination can stimulate chloroplasts, and at the same time, they are reducing equivalence in the mitochondria that can drive the mitochondrial activity. So, what are the source of reducing equivalence to mitochondria for ATP production? In the dark, of course, the TCA cycle will generate a lot of NADH, and the TCA cycle in the dark is a cycle. However, under illumination, the TCA cycle will slow down, and it is not a cycle. So, if based on our ATP data, we can say that, okay, linear electron flow did not supply enough ATP because the ratio is lower than 1.5. However, if we reverse this ratio, we can see that the linear electron flow actually generates surplus NADPH. So if plant chloroplasts cannot import ATP, that means that they need to export surplus reducing equivalent to balance the NADPH and ATP ratio. So in Chlamydomonas, it has been shown that the surplus NADPH generated by linear electron flow in the chloroplast can be converted into malate and NADP to regenerate the electron acceptor, and the reducing equivalent carried by the malate can be exported to the cytosol, which can then be imported into the mitochondrion, and this can be used to generate NADH for the mitochondrial electron transport chain to generate ATP. So this happened in algae. But how about in plants? In Arabidopsis, it has photorespiration because it has C3 photorespiration. Is it because rubisco is not always very specific to CO2? Sometimes rubiscos will pick up oxygen, and this will generate 2PG. And 2PG need to be converted back to PGA by 8 enzymatic steps in chloroplast, peroxisome, and mitochondrion. And one of the steps is the glycine decarboxylate, and this step can generate NADH. So theoretically, for mitochondrion in Arabidopsis, there could be two sources of NADH. One is the malate OAA shuttle. Another source is the glycine decarboxylation. So in fact, in this paper published 3 years ago, they proposed two models. Okay, so in model 1, both GDC and malate OAA shuttle supply NADH to the mitochondrial electron transport chain. And in model 2, the GDC generate too much NADH that exceeds the capacity of the METC, so that the surplus NADH has to be converted into malate and export to the cytosol. So in these two models, the direction of the malate OAA shuttles are opposite. This is import and this is export. In fact, there are many papers that support model 1. You can see that in this paper, the malate valve was suggested to transfer NADH or reducing equivalent from the chloroplast into mitochondria, like here, okay? And this 4 paper also support model 1. You can see that malate from chloroplast is import into mitochondria. However, there are also some paper support model 2. In model 2, they suggest that photorespiration generates more NADH than the NADH dissipating capacity of METC. So that surplus NADH is exported in the form of malate, like this. So from the mitochondrion, export to the cytosol. So question is, which model is correct? So we have adopted an NADPH sensor developed by Professor Yang Yi. This sensor is also a radiometric sensor, and it has several versions. INAP1 is a high affinity NADPH sensor, and INAP4 is a low affinity sensor, and they require a pH normalization using INAP-C. Professor Yang also generate a sensor called Sonar that can measure the ratio of NADH to NAD. And we also apply these sensors and introduce them into Arabidopsis. And the transgenic lines are normal. And we have introduced these sensors into cytosol, past it, and for NADPH sensors, also to peroxisome. Then we carry out elimination experiment, and we can see that in the cytosol, this is the NADPH sensors. In the cytosol, when they are light, it reduces. Okay, elimination reduce cytosolic NADPH, increase chloroplast NADPH. This is quite normal, but reduce NADPH in the peroxisome. And based on the response of the high affinity and low affinity sensors, we also conclude that the cytosol has lower NADPH level than chloroplast and peroxisomes. So this is the chloroplast, this is the NADPH sensors. We can see that illumination at 40 or 296 can increase stromal NADPH, but when we turn off the light, it will drop. Illumination also increases the NADH-NAD ratio, but when we turn off the light, it will drop. Then we use a AAN, which is an inhibitor of the glycine decarboxylate. We can see that AAN application, there is no increase in stromal NADPH anymore during illumination. And there is also no increase in stromal NADH-NAD. So there is no increase in reducing equivalence in the chloroplast when AAN was applied. But stromal ATP increase also still happen, and also mitochondrial matrix still happen. So under AAN treatment, there is no increase in stromal NADPH or NADH-NAD ratio. We also apply sodium hydrogen carbonate, then this will result in high CO2, and there will be no photorespiration because Rubisco does not pick up oxygen. We can see that the application of hydrogen carbonate also, we cannot see any increase in stromal NADPH or stromal NADH-NAD. So, based on this AAN and this inhibitor or hydrogen carbonate, so we show that without photorespiration, increase in stromal NADPH and NADH-NAD ratio disappear. So, based on our data, we believe that model 2 is correct. That is, the GDC actually generate a lot of NADH to supply the mitochondria electron transport chain, and then the surplus NADH is exported in the form of malate to the cytosol. Is it because if we inhibit the photorespiration by AAN, or when there's no photorespiration, there is no increase in stromal NADPH? So that means that in Arabidopsis, during photosynthesis, the surplus NADPH can be converted into malate and export from the chloroplast, but at the same time, photorespiration generate NADH, and they are too much, and the surplus NADH will also be converted into malate, and which is also export to the cytosol. This is to regenerate NADP and NAD as electron acceptors to these two important electron transport chain. So the malate will accumulate in the vacuum. In addition to our data, these are the other observations. First is that C3 Rubisco pick up oxygen and CO2 at a ratio of 1.3. That means that the photorespiration flux is also very high. And glycine decarboxylate is the most abundant soluble protein in C3 mitochondria, comprising approximately 40% of the mitochondria matrix protein. That means that there is a high demand for this enzyme. And the volume of the mesophyll stroma of spinach was calculated to be 20 times of the volume of mitochondria. That means the 2-PGA generated in the chloroplast has to be concentrated into a very small mitochondria, and then not the 2-PG, but the glycine, okay? So there will be a high production of NADH. And then it is also obvious that malate will accumulate during illumination and decrease in the dark. So now we have a better understanding on what happened in the mesophyll. Now let's look at the gut cell. So whether gut cell chloroplasts carry out photosynthesis has been a debate for decades. So these papers were published 40 years ago. At that time, they conclude that gut cell chloroplasts cannot fix CO2. But later, a paper report that gut cell chloroplasts can fix CO2. And in this paper published in the annual review of plant biology in 2020, and it showed this diagram. In this diagram, you can see that the chloroplast can receive light and then can carry out CO2, and also the chloroplast can export ATP. Okay, chloroplast GC photosynthesis can export ATP. This ATP supply ATP for the proton ATPase pump, that is to pump out proton to activate the K channel. So this was the belief until 2020. So they believe that upon elimination, gut cell chloroplast could export ATP to the cytosol to support H pumping during stomata opening. So a lot of paper cite that. But if we look at our ATP sensor line, we can also look at the gut cell. We can compare gut cells and mesophyll. And this is the cytosol. This is the gut cell cytosol. This is the gut cell chloroplast ATP level. And we can see that in the gut cell, the cytosolic ATP level is higher than chloroplast. Okay, these are the 3 time points. Okay, end of night, 2 hours after turn on the light, 8 hours turn on the light, and then these are higher. So it's unlikely for gut cell chloroplast to export ATP to cytosol because cytosolic ATP is higher than in the stoma. Then we also find that using the same plant, elimination can only induce stomal ATP increase in mesophyll, but not in gut cell. Elimination can only increase NADPH in mesophyll, but not in gut cell. And for stromal pH, it is increased, alkaline, in mesophyll by elimination, but not in gut cell. So it seems that elimination did not cause an increase to the stromal ATP and stromal NADPH in gut cell. To look at this carefully, we have isolated protoplasts from mesophylls and gut cell. And then we carry out Western blotting by eco-loading using the actin as a marker. And we find that in the gut cell protoplast, it has much lower protein concentration of photosystem, lower concentration of Rubisco, CPP enzymes, or enzyme for sucrose synthesis and starch synthesis. But it has comparable amount of Pep-C and MDH compared to the mesophyll. So Pep-C and MDH, okay, Pep-C actually can fix CO2 into OAA, and MDH can convert OAA into malate. And then we have also titrate the amount of Rubisco of gut cell protoplast and mesophyll cell protoplast, and we find that in the gut cell protoplast, its Rubisco is about 1/16 of mesophyll by protein weight. Okay, whereas the starch synthesis enzyme AGPase is 1/6 of the mesophyll by titration. In fact, many years ago, it has been published that using the tulips epidermis, which is believed to have a high amount of gut cell, is that under light, the CO2 is mainly fixed into malate rather than free PGA. It can still fix a little bit amount of free PGA like this. Okay, so free PGA production requires this, but because the protein level are low, so it generates only small amount. Whereas in the dark, okay, this is in the dark, it can still fix CO2, but mainly in the form of malate. And if you look at mesophyll, this is gut cell, okay? If you look at mesophyll, we can see that the mesophyll in the light, okay, can mainly fix CO2 in PGA and sugar, and a very small amount of malate. But mesophyll in the dark mainly can fix CO2 in Mali, and this is the product of PEPC and MPH. So you can see this is high, and this correlate with the high concentration of PEPC and MTH4 in the gut cell and in the mesophyll. And this does not require light to fix CO2, but this requires light to fix CO2. So if the gut cell cannot or does not generate a lot of ATP by photosystem, then it has to be import ATP from the cytosol. We look at our gut data, we can show that the gut cell actually express a high concentration of NTT1. And then we isolate gut cell chloroplast and we add ATP, and we can see that ATP can get into the gut cell protoplast and increase the sensor signal. Whereas for chloroplast, there's no increase. So in gut cell, mitochondria generate cytosolic ATP, and then through the NTT1, ATP is imported into the stroma of the gut cell. And if we compare the starch metabolism of mesophyll and gut cell, we can see that they are different. In mesophyll, the mesophyll accumulates starch, and in the dark, it will break down the starch. However, in the gut cell, in the dark end of night, it still have some starch. And in the first hour, when the light is turned on, the starch will break down. Okay, you can see starch increase here. But for the gut cell, in the first hour, the starch will break down, and then it will be synthesized. Okay, so this is very different from the mesophyll. And so what happened here? Why starch break down and increase? And what are the energy and carbon source for gut cell? So our collaborator have developed a method to look at the starch granules in the gut cell. And this is the wild type plant, and this is end of night. So end of night, they are still starch, like here, okay? And 1 hour disappear. So 1 hour, or here we use 2 hours, you can see starch granule disappear, and the light turn, prolonged illumination, the starch synthesized again. However, in the NTT1 mutant, we do not see a lot of starch granule here, no starch granule, and even 8-hour light, okay, there is a very few starch granule here. And NTT2 also have a lower amount of starch granule. So that means that the importation of ATP from the cytosol is important for starch synthesis in the gut cell. And lack of NTT1 also hamper stomata opening. Okay, so this is NTT1 mutant. And then, so based on our data, we propose a model. So at dawn, when there are starch in the gut cell, and when mesophyll has not yet generated enough sugar for the gut cell need to degrade the starch so as to generate sugars to increase the osmotic pressure for stomata opening. The hexose can also undergo glycolysis as well as generate PEP. This allows the fixation of CO2 to generate OAA and then the MALE. Okay, and then the PEP and MALE can also provide NADH to the TCA to generate ATP. And this cytosolic ATP can be used as an energy source for the proton pump. And this will then activate the input of the potassium and then also other ion channel because it changes membrane potential, and then also input of MALE, and then this will increase the osmotic pressure of the gut cell, and that helps stomata opening. Okay, so if there is no NTT1, okay, there is no starch. Okay, if no NTT1, no starch, so the opening will be hampered. Okay, no starch, then this opening would be hampered. And then when the starch disappears, then the gut cell need to synthesize starch. So then after 1 hour, the mesophyll can fix a lot of sucrose, export sucrose, and then the sucrose can be used as a carbon source and energy source for gut cells. And then not only it generate and sugar, the hexose can generate G6P, which can be input into the gut cell chloroplast as a carbon skeleton. And at the same time, ATP generated from the mitochondrion can be input into the gut cell gut cell chloroplast, and then the ATP and the carbon can help starch synthesis. So if there is no NTT, there will be much less starch synthesis. So, based on our data, we conclude that gut cell can still carry out photosynthesis. Not only our data, but also this data, okay, a little bit, free PGA, but this is not the major pathway. So if we compare gut cell and mesophyll cell, actually the gut cell is much smaller. Its volume, its chloroplast volume is only 1 out of 16. So you can see that this is a mesophyll protoplast. This is a gut cell protoplast, which is much smaller. And if we look at the chloroplast diameter, gut cell chloroplast diameter is about 2 micron, and for mesophyll chloroplast is about 5. So if we calculate the volume, this is about 1 out of 16. The riboscope content is also 1 out of 16 in terms of protein amount, whereas for Pep-C content, those cell types are comparable. So in mesophyll, the major CO2 fixation pathway is riboscope, whereas in the gut cell, the major CO2 fixation pathway is Pep-C. And mature mesophyll cell does not express NDT transporter, whereas gut cell express NDT transporter. And the major source of stromal ATP in gut cell is importation from from cytosol, whereas for mesophyll, well, during the light, it's a photosystem, and during the dark, it is the parasitic glycolysis. So so I would like to thank my collaborator in the gut cell project, Dr. Santalia of ETH, and also the fundings. And I would also like to thank my other collaborators, such as Marcus and Wayne and Per in the ATP sensor project. And of course, I have to thank my students and postdoc. And my university also provide pretty good postdoc fellowship and PhD scholarship. And so if you are interested in our work, and please write to me. Thank you very much.
Wow, great presentation. Really nice work. So now we have we really have time open for questions. Maybe I start with a question. Maybe I missed it. I should know. So in the mature chloroplast, there's no import of ATP, certainly during the day, right? But you in one slide you mentioned there is large consumption of ATP at night, and chloroplast consume large